Welcome back to the playlist on our biosignaling pathways. If I seem a little out of it in this video, it's about 11:30 at night, and I just recently took 2 benzodiazepines, basically some Xanax to help me sleep, and I was having trouble sleeping, so I decided to do this instead. Okay, but suffice to say, in this video, what we're going to end up doing is we're going to look at the various ways to ultimately terminate a cyclic AMP-mediated response. Okay, so in a previous video, what we looked at is for instance, if our example of a hormone that bound to a receptor was epinephrine. Okay, epinephrine, right? And it bound to the adrenergic receptor, which is shown here, right? And you ultimately had an alpha subunit of the G protein. So this right here is our G protein. And in the presence of GTP bound to the alpha subunit and the hormone bound to the adrenergic receptor, the G protein dissociated over this way, bound to another enzyme, in this case it's adenylate cyclase, and catalyzed the irreversible conversion of ATP to cyclic AMP, right? And as we increase the concentration of cyclic AMP, we ultimately, through a mechanism that we've already looked at, we increase the activity of protein kinase A, and ultimately through protein kinase A, we attach phosphate groups to certain proteins that activate a biological response. Okay? So what we're going to look at in this video is, yeah, it's good and all that we can do that, but how do we terminate the response? And in fact, there's a lot of ways that ultimately work in tandem to terminate the response. And some of them are more obvious than others. For instance, the first one we'll look at that's actually very obvious is, well, first of all, the hormone dissociates from the actual receptor, right? That's our first way, okay? That one seems really obvious, but if you think about it, it makes sense, right? The only way you can have that alpha subunit dissociate from the regulatory component of the G protein is if the hormone's bound to the receptor. Now, it is true you have some constitutive activity even without the hormone, but in general, once the hormone dissociates, or any agonist, once it dissociates, the alpha subunit recombines with the regulatory subunit of the G protein, and that effectively helps to terminate the response. So that one's sort of obvious, okay? What else can we do? Well, one of the main ways, and actually one of the most important ways, is through something called a phosphodiesterase. Well, what's that? Well, for handiness, I've drawn the structure of cyclic AMP over here, and what we're going to look at in this part of the video is something called a phosphodiesterase. And all phosphodiesterases do is they hydrolyze phosphodiester bonds. Well, if you look here, okay, this right here is our phosphodiester bond, okay? So what we're going to have is a hydrolysis of this bond, okay? And we'll find out which part of the bond it is, okay? Now, in general, phosphodiesterases are scattered all over the cell. There are those that degrade DNA. For instance, if you've done any kind of biology lab, you've probably worked with DNases, you've heard of RNases. Those are phosphodiesterases, but they're specific for sort of, as you could consider, linear chain phosphodiesterases. As you can see here, this phosphodiester bond is cyclic. Okay, so it turns out that these phosphodiesterases are specific for cyclic nucleoside monophosphates, and specifically this one that we're talking about here is specific for 3' 5' cyclic AMP. Okay, and notice I specify here the 3' 5', so this is our 3' carbon, and this right here, this one is our 5' carbon. Okay, so this particular phosphodiesterase is going to hydrolyze the bond between those with respect to the phosphorus. Okay, so effectively what this enzyme does, okay, it's more complex than this, but it effectively allows water into the active site, and you get a nucleophilic attack on that phosphorus. These electrons kick up to generate a trigonal bipyramidal intermediate. The electrons kick back down, and the leaving group is your 3' hydroxyl group, and it comes out and abstracts a proton. So what you end up generating is normal, you generate a normal 5' AMP. Okay, so using this 3' 5' cyclic AMP phosphodiesterase, we end up generating 5' AMP. And if you hear a sound in the background, that's just my washer. I'm washing some clothes right now, so don't worry about it. But anyways, we hydrolyze this bond right here, and we end up generating 5' adenosine monophosphate. Okay. Now, Adenosine monophosphate doesn't have the same activity that cyclic AMP has. Cyclic AMP is able to activate protein kinase A, right? It's able to bind to those regulatory subunits that are attached to protein kinase A anchoring protein, right? And it's able to bind to those regulatory subunits to cause dissociation and activation of pKa, right? But adenosine monophosphate doesn't have that same activity. So by hydrolyzing that phosphodiester bond, you terminate the activity of cyclic AMP by converting it to a non-active AMP. Okay? That's one way to do it. Okay? Another way that we can do it is recall that in the process of activating pKa, protein kinase A, that enzyme attached phosphates to specific residues. Usually they're serine or threonine residues. In this case, we have a serine. Now, I can put a generic R group here and say that if R is hydrogen, it's serine. If R is a methyl group, then it's threonine, right? But in general, protein kinase A and related kinases are attaching phosphates to serine and threonine. Now, what you can do is you can instead of having activated kinases, eventually what you'll have is activated phosphatases, okay? So again, in a very similar mechanism to what we saw in the phosphodiesterase, we have phosphatases that allow water into the active site. Now, phosphatases are different than phosphodiesterases in that phosphodiesterases hydrolyze phosphodiester bonds. Effectively, what a phosphatase is doing is just cleaving off a non-cyclic phosphate, okay, like this one. So water is allowed into the active site. You get a nucleophilic attack here. Electrons kick up to generate a trigonal bipyramidal intermediate. Electrons kick back down, reform the pi bond, and this bond comes out and abstracts a proton. And effectively, what you get is the protein. So you'll get this. You'll get the protein back, right? But instead, it will just have the hydroxyl group, and depending on whether it's threonine or serine, it'll have a methyl group or a proton, right? Does that make sense? Okay. So phosphatases, in other words, if kinases activate the pathway, the biosignaling pathway downstream, phosphatases will reverse that by inactivating it. Okay? What's another way we can do this? Well, there's even another way. Okay? Remember that in activated G proteins, the G protein has bound GTP. So in other words, this bound GTP, okay, this bound GTP is absolutely required for the activity of the G protein, right, the alpha subunit. Well, it turns out that the alpha subunit has intrinsic GTPase activity. In other words, the alpha subunit is a GTPase. What that means is that it's a GTP hydrolase. So if you have GTP, okay, so in other words, if you have this, so here's guanosine, and then you have 3 phosphates, right? Here's GTP. It's just an abbreviated version, right? Effectively, what's going to happen is the enzyme GTPase, which is part of the G protein, is going to allow water into the active site, and it's going to do nucleophilic acyl substitution, and GDP is going to be your leaving group, okay? GDP will remain attached to the alpha subunit, but the whole point is that the G protein alpha subunit has intrinsic GTPase activity. And so every single time the G protein alpha subunit binds to adenylate cyclase, of course adenylate cyclase becomes activated, but at some period of time, once the alpha subunit has bound to adenylate cyclase, the G protein GTPase activity will hydrolyze the GTP to GDP, and as soon as the GDP is generated, it remains bound to the alpha subunit, and with GDP bound, the G protein dissociates back to this point where it's bound to the regulatory subunit. So when we initially look at a G protein, assuming GTP is not bound and GDP is bound, you'll ordinarily see it at this point attached over here to these regulatory subunits. Okay? So the whole point is if GTP is bound, you're going to see it right there. If, for instance, GDP is bound, it's going to be bound to the regulatory subunit. So, in other words, to get the alpha subunit back to the adrenergic receptor and the regulatory subunits of the G protein, beta and gamma, you have to hydrolyze the GTP, and it turns out that the alpha subunit has intrinsic GTPase activity. Okay? And effectively what you would see is, and I challenge you to draw it, draw GTP and then do a nucleophilic acyl substitution on the gamma phosphate. Okay? and you'll end up generating GDP, and that remains attached to the enzyme. Phosphate dissociates. But the point is that you hydrolyze the GTP, and it goes back to GDP and causes the alpha subunit to move from adenylate cyclase back towards the regulatory subunits. Okay? And in general, those are sort of the simplistic ways to terminate a response. Now, there is something that another thing we refer to, and I have a whole other picture for it, and this is what we call desensitization. Let me write that. This is desensitization. Okay? Desensitization is the process by which, even in the presence of a hormone, the response to the hormone decreases. Okay? That's sort of an overly simplified definition of it. So, if you ever heard of the concept of adrenaline junkies, right? Remember, we're dealing with adrenaline, epinephrine, so this will be a perfect example. When you have with an adrenaline junkie, they do some kind of stunt that basically causes an increase in extracellular epinephrine, right? And so epinephrine's going through and it's binding to the receptors, right? But over time, remember that the adrenaline rush goes away. And part of the reason the adrenaline rush goes away is because of desensitization, okay? And desensitization in general for these G protein coupled receptors is catalyzed by an enzyme called beta-arrestin receptor kinase, okay? So the enzyme, I'm going to write it over here, beta-arrestin receptor kinase, okay? And so this is typically abbreviated BAARK. Now this is not BARK, it's beta-ARK. This is a beta, right? But essentially what beta-ARK, what beta-arrestin receptor kinase is going to do is it's going to phosphorylate the adrenergic receptor. Okay, so in our example, right? Our example is the adrenergic receptor. So it's going to phosphorylate the receptor. Okay? Hope that makes sense. So it's beta-arrestin receptor kinase. It's phosphorylating the receptor. And the reason it's doing that is because the phosphates on the adrenergic receptor have specificity for a protein called beta-arrestin. And that's the beta-arrestin in this name. Okay? So what's going to happen is you're going to have beta-arrestin, okay, that's going to come over here and it's going to bind to the phosphate. And specifically the phosphate is going to be on serine residues. So we can consider this a serine residue. And this protein right here is called beta-arrestin. Okay? Now beta-arrestin has two main functions. Okay? Number one, beta-arrestin interacts with the adrenergic receptor on phosphorylated serine residues. And effectively what happens when beta-arrestin binds is it sort of, you could think of it as it destabilizes the interaction between the G protein regulatory subunits, well, the G protein in general and the adrenergic receptor. So if you destabilize that interaction, what happens? Effectively, your G protein dissociates, right? So effectively what beta-arrestin does is it causes the G protein to dissociate. So even in the absence of the regulatory subunits, the alpha subunit loses its function. And so ultimately, if you don't have a G protein attached to the adrenergic receptor, even in the presence of a hormone like adrenaline, right? Even in the presence, there's no activity, right? Because the G protein has dissociated. And effectively, that's done by beta-arrestin. Beta-arrestin causes destabilization of the interaction between the adrenergic receptor and the G protein, okay? With no G protein, you have no response, okay? So I hope that makes sense. Now, there's one other thing that beta-arrestin does, and I'm not going to draw a picture for this because it's too complex, but I hope you can picture this in your mind. You have your membrane of the cell, right? And actually, I might go ahead and draw this. Let's sort of come over here. I'm going to sort of draw the membrane like this. Okay, here's the membrane, right? And maybe we can draw a few adrenergic receptors in here. So here's an adrenergic receptor. Here's another one, right? The other thing that beta-arrestin does is it catalyzes the invagination of the cell membrane with the inclusion of the adrenergic receptor. So what you'll end up getting is something that looks like this. So you'll get sort of a vesicle type thing, okay? And in the membrane of this vesicle, there are adrenergic receptors. So effectively, so it'll look like this. Okay? So how does this happen? Well, the membrane invaginates. So you have to picture this as the extracellular fluid. Here's the cytosol. So it effectively pulls the membrane inside the cell in the vesicle. But the thing is that these adrenergic receptors are bound in the vesicle membrane. So effectively what happens is now you have the adrenergic receptors pulled out of the membrane, and so if there's less adrenergic receptors, even in the presence of the same concentration of adrenaline, now you have less of a response because there's fewer receptors, right? Okay, so I hope that makes sense, and that's catalyzed by beta-arrestin. Now the actual phosphorylation of serine residues right here is catalyzed by beta-arrestin receptor kinase, but the two molecules tend to go together, okay? So in general, that's your ways of reducing the response to a G protein coupled receptor and cyclic AMP. So let's do a quick recap. So remember that in order for the G protein coupled receptor to be activated, you have to have, number one, you have to have the hormone bound to the receptor, and you have to have GTP bound to the alpha subunit of the G protein. Okay? Assuming those two things occur, alpha subunit dissociates and interacts with adenylate cyclase. Okay? And of course, then you get the conversion of ATP to cyclic AMP with the loss of pyrophosphate. Pyrophosphate gets cleaved, hydrolyzed by inorganic pyrophosphatase. You get a driving force by the release of free energy through the exergonic nature of the reaction, negative delta G. Right? You get an increase in activity of protein kinase A, which phosphorylates certain proteins, causing them to become activated. Right? Now, remember, our first way of causing the response to be terminated is just the hormone dissociating from the receptor, right? And that seems kind of obvious, but it's something that's important to remember. Remember that a molecule being bound to a receptor is only a probability function, okay? Of course, it's dependent on the affinity constant for the receptor, but it's just a probability. It's not an indefinite thing. Hormones and neurotransmitters are constantly associating and dissociating, okay? And of course, if you dissociate the hormone, you can't have an active G protein. Okay. The next way we saw was to actually, first we did what? We used the phosphodiesterase, and that's a simple hydrolase, right? It's a hydrolase that cleaves the phosphodiester bond of 3' 5' cyclic AMP, generating 5' adenosine monophosphate, which has none of the activity that cyclic AMP has, thus terminating the response. Okay. The next way that we saw was to actually use a phosphatase to cleave off phosphates, usually from serine and threonine residues of proteins. If the phosphate addition activated these biosignaling proteins downstream, then cleaving the phosphate off would deactivate them. Okay? The next thing we saw was the intrinsic GTPase activity of the G protein. So at some time after the point that the alpha subunit interacts with adenylate cyclase, of course, adenylate cyclase becomes activated, but the alpha subunit will hydrolyze GTP to GDP in a simple nucleophilic acyl substitution mechanism that generates GDP, which remains bound to the enzyme, and of course, then you have the alpha subunit dissociates back and interacts with the regulatory subunits over here. Okay. So that terminates the response. If you don't have the alpha subunit bound to adenylate cyclase, there's no response. And just remember, this enzyme right here, it doesn't have to be adenylate cyclase. There are other enzymes that it can interact with. It's just that for the purpose of examples, we usually use adenylate cyclase because conceptually it's easy to understand. Okay? So that's that. The last thing we talked about was desensitization, which remember is basically a process by which even with the same concentration of hormone, or neurotransmitter in the extracellular fluid, the actual magnitude of the response is decreased because of some mechanism inside the cell. Our mechanism is catalyzed by beta-arrestin receptor kinase, this guy right here, and he phosphorylates the adrenergic receptor at specific serine residues, right? And so then beta-arrestin, which is a protein, associates with those serine phosphates, right? And it destabilizes the interaction between the G protein and the receptor, and thus the G protein dissociates, right? G protein dissociates, and without the G protein, you have no response, right? The other thing that beta-arrestin does is over here, where it causes invagination of the membrane at areas where you have the adrenergic receptor. And so if you pull the adrenergic receptor inside the cell, then even in the same concentration of hormone outside the cell, there's not as much of a response because there's fewer adrenergic receptors. Okay, so I hope this video gave you a little bit of intuition on how we terminate a cyclic AMP/G protein mediated response. If you have any questions, just make a comment below the video. See you soon.